coolest techniques that you likely haven't heard of is called hydrogen-deuterium exchange mass spectrometry, or HDX-MS. It works a bit like putting a protein or other molecule in a bath and seeing which parts get wettest fastest. And the way that this is done is by using heavy water in this bath. And so this water has deuterium instead of protium. So basically it has a version of hydrogen that is heavier. It has an extra neutron, but it reacts just like normal hydrogen would. So it's not radioactive or anything like this, but it is heavier. And so therefore, if proteins get this incorporated into them, then those proteins in those places where the deuterium was incorporated are going to be heavier. And so if we can see which parts of the protein gets kind of labeled by this heavier water, that is, which parts of the protein are kind of getting wetter in the bath, we can then see which parts of the protein are exposed to the solvent, so which are on the surface, say, or in a channel, which of them are free from interactions with other molecules, and which of them are going to be free from strong secondary structures, things like alpha helices and beta strands. All of those things would prevent the protein from getting wet in that spot. That is, they would prevent it from exchanging their backbone protiums, the normal hydrogens, for deuterium, this heavy hydrogen. The way that it works is by taking advantage of the protein's amide backbones. So in the backbone of proteins, they have these amides, which is basically an oxygen double bonded to carbon, and then that carbon is attached to a nitrogen, and that nitrogen has a hydrogen. Now, the hydrogen that's attached to that nitrogen can actually exchange. And so what this means is basically we can have that hydrogen being replaced. And not only can it be replaced with a normal version of hydrogen, so a protium, which has 0 neutrons and 1 proton, but it can also be exchanged for a deuterium, which has 1 proton and 1 neutron, and thus a weight of 2 instead of 1. And so what we can do is we give this protein a bath in this heavy water, which contains this deuterium instead of the protium. And now if that hydrogen in the amide backbone gets exchanged, it's going to get exchanged for this deuterium. And therefore the region of the protein that got that exchanging is going to get heavier. And so what we do is we give this protein a bath in this heavy water, and then we take samples over time and we quench them. What we mean by quenching them is we basically stop any of the exchange. The way that we can do this is by sticking them under low pH conditions and cold temperatures where we're not going to get that exchange occurring, and we switch them back into that normal water so that any exchange that does occur is going to then be back into water and isn't going to continue labeling after we want to quench the labeling. We do this at different time points, and then what we do is we kind of cut up the protein to get different peptides. We use liquid chromatography in order to separate the peptides, and then we use mass spectrometry. And so MS, so you might see liquid chromatography LC, and then mass spectrometry MS, so LC-MS, and that is then going to allow you to get the mass to charge ratio of those peptides. And then you can map those to databases to see where in the protein that corresponds to. And if you have a structure of the protein, one of those static structures, you can map it to that structure, or you can map it to the predicted structure. You can then look over time and see which regions of the protein are getting heavier faster. And those regions of the protein that get heavier faster are going to be regions that are likely solvent exposed, that are free from those strong secondary structures which would tie up the amide hydrogens. So when you're making those alpha helices, when you're making those beta pleated sheets, those are held together by hydrogen bonds involving that very hydrogen that you are exchanging for the deuterium. And so if they're tied up, then you're not going to see the exchange as rapidly. You're also not going to see the exchange at a part where the molecule is interacting with another molecule. And so what you could do is you can look and see which regions were protected, which were unprotected. Something you might be wondering is, yeah, I get that there are regions that are flexible, but isn't a lot of the protein kind of tied up in that secondary structure all the time? The answer is yes, but proteins breathe. And so we can think about proteins as undergoing conformational changes. So we have these, often think about like these broad changes, these big shape changes of proteins. But proteins also undergo very local changes, and they kind of breathe in their backbone without kind of coming apart. And so if you think about 2 strips of Velcro, you can have regions of the Velcro where like a couple of the hooks come apart, and overall, the strips of Velcro still stay together, so your shoes aren't like coming apart or whatever, but you have this local region where that local region is now available to get wet. And so when the protein backbone breathes, you kind of get hydrogen bonds that are holding together those secondary structures and things, they can kind of break for a second. And when they break, then the deuterium has a chance to sneak in if you're in deuterated water. But it's going to be harder for this to happen. You're going to have less chances for this to happen if that region is very tied up in the secondary structures, or if it's tightly bound to another molecule. And this is why you can have it be that you get this uptake over time. And so all of the protein is basically going to get to that maximum labeling eventually, but that eventually is going to take longer for regions of the protein in which that hydrogen in the backbone is tied up. And you also might be wondering, well, what about hydrogens in the side chains? What about hydrogens attached to carbons? The ones attached to the carbons are just going to take way too long to exchange. As for the side chains, well, those are going to exchange back to the normal water before you can actually quench it, and so you're not even going to see that they did get heavier instantaneously. And so it's those amide backbone ones that you're going to see. There are different types of experiments that you can do to ask different questions. If you just want kind of an idea about the overall protein flexibility, availability, all this stuff in different conditions, then you don't even need to cut up the protein. You can just do what's called global. If you want information at the peptide level, now we're getting local. You can do this over time to get information about how regions of a protein might be changing over time, or which regions of the protein might be more susceptible to exchange. And the regions susceptible to exchange are going to vary if you have a binding partner or ligand. And so if you want to see where a ligand is binding, one of the ways that you could do this is actually by using HDX-MS and using differential HDX-MS in order to kind of test under different conditions and see if I have a ligand present versus if it's not present, which regions, which peptides show stronger protection in the presence of the ligand than the absence. And that gives you information that that region might be making an interaction with that molecule. Of course, you need to be careful because you can also just have kind of allosteric changes where you have kind of broader structural changes that are happening even though the molecule is binding somewhere else. But here is where kind of the peptide level resolution comes in handy, because if you see a change happening in just this part, but not other parts, that's kind of strong indication that you have something locally happening. One of the reasons why I wanted to do this post was I think that HDX-MS will have growing importance in the coming years because it's able to offer information that you can't get from other experimental methods, or even prediction methods like AlphaFolds. X-crystallography and cryo-EM are great for giving you static snapshots of proteins, and accurate ones. AlphaFold is great for giving you predictions of static snapshots of proteins. You can use techniques like molecular dynamics in order to model the movement of proteins and to do molecular docking to see where molecules might bind, but those are just computational methods. And so for all these things, you're going to want some sort of confirmatory information, some complementary information that you can get from another technique. A technique that's going to give you information about the protein as it's moving around dynamically and interacting with other things. And HDX-MS is one of the ways that you can get this information. It's not giving you the level of resolution as you would get with something like cryo-EM or X-ray crystallography. The kind of highest level information you can get is at the peptide level. Well, actually, if you have overlapping peptides, you can get information more at the residue level, but you can also have artifacts or different problems. And so each technique has its own pros and cons, but HDX-MS is able to offer this information about the dynamic nature of a protein, the flexibility of a protein. It can offer you information about regions of a protein that don't have a set secondary structure, so regions that are intrinsically disordered, and IDRs, which might have been hidden in cryo-EM or X-ray crystallography because they're kind of blurred out, or the AlphaFold are those regions that are all spaghetti-like and they're like bright orange, are like, don't trust me. Well, that's because those regions are moving around a lot. With HDX-MS, you can actually see kind of if those regions might get more protected under certain conditions, which could indicate that you're undergoing some sort of structural change there, or that you're binding to another molecule. So all this valuable information that you can get from a tool like HDX-MS. So again, the basic idea is that you take a protein and you stick it in this deuterated water, you take time points. At these different time points, you quench the samples, and so you take a sample of your protein and then you stick it into low pH cold conditions in normal water, and this is going to stop any further exchange for the deuterium. And then you do this over time, and you take those pieces that you got, and the pieces that were flexible, the pieces that were solvent exposed, that weren't bound to something else, these are going to get heavier faster. And how do you know if they get heavier? Well, you use mass spectrometry, so you typically use LC-MS, so liquid chromatography mass spectrometry. You cut up the protein into peptides, and then you send those peptides traveling. You separate them a bit by this liquid chromatography step, and then you separate them by their mass to charge ratio. You get those mass to charge ratios, and then you map that to a database to see kind of which regions of the protein were getting heavier or lighter over time. You can then do peptide uptake plots where you look at the uptake of specific peptides over time, or you can kind of plot those onto a structural model, either one that was kind of experimentally solved with X-ray crystallography or cryo-EM, or a predictive model such as you got from AlphaFold. And then you can see kind of which regions of a protein have this higher or lower uptake. You might also see kind of the whole sequence shown, and then you'll see kind of which peptides show more or less protection under various conditions or at different time points. So various ways you might see this information displayed. You might see it as a difference under different conditions. And so you just need to look at kind of like the legend and the key and the guide to see what's being displayed how. And you can get valuable information about the protein structure. I actually used it in my grad school work, and so with the collaborator who actually collected the data, but I got the sample, he collected the data, I analyzed the data, he helped me analyze the data, so it was a lot of fun, and we got some really important information. 0 minutes, this is kind of our baseline. We don't see any deuterium uptake. At half a minute, we start to see our envelope moving over. Keeps moving, it keeps moving, and it keeps moving, and we can plot these onto a peptide uptake plot to give us the deuterium uptake over time. Because all those peptides have different sizes and different amounts of hydrogens, it's useful to convert this to a fractional uptake rather than just a raw uptake. And this is going to allow for a more easy comparison. So you can see that the maximum amount of uptake we get after 5 minutes is about 50 to 60%, or 0.5 to 0.6 fractional uptake. We can then plot these onto the structure, the crystal structure of the Argonaut protein. Here we're using a colorblind safe color scheme, and so purple is going to be your most protected, so the least exchange, and yellow is going to be the least protected, the most exchange. Sometimes you might see a blue to red scale, with blue the most protected and red the least protected. But in this case, we're using this colorblind safe scale, and so here you can see you're kind of a middle amount of protection, which makes sense because you kind of have some of this hydrogen bonding, keeping the secondary structure of the alpha helix. If you look at this more flexible region, however, well, here you see you have a really high uptake. And so you have almost full uptake in the fragment after 5 minutes. And even at 30 seconds, you already have about 70% uptake. Whereas in regions that have a really strong secondary structure, maybe are a little more solvent protected, maybe a little more central in the protein, these are going to have a very low exchange. And so you can see that this fragment here is going to be very well protected. You can do this for all the peptides on the protein that you have coverage for. And again, you're not going to see as high of exchange if a region of a protein is kind of well protected from the exchange, if it's not getting wet, which can happen if it's tied up in strong secondary structures, things like alpha helices and beta pleated sheets. If it is kind of hidden in the center of a protein, so it's not solvent exposed, if it is bound to a ligand, so another molecule, and that sort of thing. And so this provides all this information about a protein under different conditions, but conditions that are kind of more realistic, or conditions that at least allow the protein to go about its normal business without you asking it to, like, hey, freeze for a picture. So I think it'll offer complementary information in the years to come. And if you want to know more about it, I got a longer video, but I just wanted to give people the basic idea of how this technique works here, because I think it often is overlooked, and so it takes kind of some specialized equipment to do it, so it's not very well used, and there are a bunch of limitations and stuff, and it's harder if your protein is more complicated and bigger and all these things, but it is a valuable technique, and I think that it's one that's worth learning about.